framework for prioritizing between interventions, impact, cost of interventions. Because if you are thinking of any intervention, then the countries need to know what will be the cost of this intervention. So that also needs to be studied. Then implementation in low and middle income countries, social, cultural, behavioral insights, tailored interventions, immunizations. Dr. Nagendra talked about the effect of vaccine on AMR, and surveillance for action. And then advocate. Once you have the evidence, you may make interventions. So advocate for prioritization of AMR mitigation. So you need to control how to minimize AMR. As we have seen, it's not possible to totally eliminate AMR, but you are only thinking of mitigating or reducing AMR by various approaches. So improve understanding of the social and economic impact of AMR over the One Health spectrum, value of investing in AMR mitigation, socio-economic burden of AMR, policy enablers, economic evaluation, sustainability enablers, surveillance informing policy. So this is the strategic objective of One Health priority research agenda for the countries. Now under this research agenda, what are the main things that need to be addressed? So there are 5 pillars of this high priority research agenda for AMR. One is on transmission, the second one is on integrated surveillance, and the third one is on interventions, fourth one on behavioral insights and change, and then the fifth one on economics and policy. So cutting across, you have gender, vulnerable populations, and sustainability. So all these aspects are considered while developing a research agenda for AMR and its mitigation. This quadripartite organization. So highest priority research areas. So in terms of transmission, so the pillar is transmission. So under transmission there are 2 high priority areas. One is to understand to what extent do various infection prevention control practices. Now we had talks on infection prevention control in hospitals, in veterinary practice, and also this similar thing has to be practiced in aquaculture, agriculture. So to what extent do these practices in One Health settings impact the development and circulation of AMR? I think this was very well touched upon by Dr. Nagendra Hegde when he talked about the impact. Is there evidence that if you minimize the use of antimicrobials or reduce the need for antimicrobials, does the AMR come down? So we need to evaluate the infection control practices and see whether they require any modifications. How can we improve the current practices based on the assessment of the impact, development and circulation of AMR in One Health sector. The second high priority area is what impacts the transmission. So what facilitates the transmission of the resistance from one organism to another? Horizontal transmission is well known. At least in bacteria it is well known. So what impacts this transmission between also humans, animals, plants and environment? So now we know that many of the resistant organisms that affect humans may be coming from other sectors. Maybe the impact of use of antimicrobials in livestock or poultry or other sectors. So what impacts the transmission between humans, animals, plants and environment, with a focus on conditions relevant to low and middle income countries. So of course this is one of the major considerations for any UN organizations. You can advise many things, but can the low and middle income countries implement these practices? Then under integrated surveillance. Now integrated surveillance was touched upon by other speakers during the first day. Dr. Anu Sharma talked about the work WHO is doing in terms of integrated surveillance, and in terms of international standard setting bodies, we have the Codex Alimentarius Commission, which has a guideline on how to conduct integrated surveillance between different sectors. So what are optimum strategies and minimum standards for adequate laboratory and human resource capacity? I will be touching upon some of these aspects a little later, because when you want to determine resistance, so what are the laboratory methods that you use and what are the constraints that you face? to establish and maintain quality of integrated AMR surveillance system at scale. Then how can existing AMR and AMU surveillance data from humans, animals, plants and environment be meaningfully triangulated or integrated to allow early detection of development, escalation or circulation of resistance across health sectors. So these are the major research priorities in terms of integrated surveillance. Now in terms of interventions, what do we need to do? So how can One Health intervention that have proven impactful for AMR control and mitigation most effectively be translated and scaled up? So infection practices may be developed at different levels. Yesterday Dr. Vandana mentioned that of course in control practice may be different. It may be at hospital level or at regional level or at national level. So how can these interventions that have proven impactful be most effectively be translated? So can we learn from the practices in one of these areas or one country or one area and scaled up in different contexts or different resource settings? And what challenges exist in systematic collection and analysis of data for risk assessment and intervention for impact assessment? So this is where the databases that are created by different agencies like by the WHONET or WAHIS or other international databases help you to gain understanding of what is happening in other countries and what interventions may be impactful. So epidemiological, economic, social in low and middle income countries. And then behavioral insights and change. So how can structural challenges and barriers to change behavior related to AMR be identified, characterized and assessed in different socioeconomic contexts? So this is something that needs to be addressed. And what strategies can be used to adapt effective behavioral interventions? Immunizations. Now immunizations we have been hearing sometimes there is a negative thinking about immunization. Even during COVID, we saw some people think that it's not necessary to immunize. And even in developed countries, now we have recurrence of diseases like measles, diphtheria in many of the developed countries because they are against vaccination. So how can we make changes? It's behavioral changes, behavioral interventions, like for immunization from one context to another, Africa, Asia, from rural to urban, or human prescriber to veterinarians. And then economics and policy. What would a One Health AMR socio-economic impact assessment based on accurate, cost-effectively collected data, that harmonized methodology and indicators in low resource setting optimally look like? And how can governments identify, prioritize and institutionalize the most relevant cross-cutting sector-specific AMR policy options? So when complex issues come before governments, they always try to avoid taking any action because there is no clear-cut way how to proceed. So we need to produce this kind of evidence to the government to develop a policy so that most relevant cross-cutting sector-specific AMR policy options can be developed, and regulatory frameworks. So regulatory framework in terms of antimicrobial approvals or permission to use, prescriptions, sale, so all these aspects related to regulation, and financing strategies to sustainably tackle AMR across One Health sectors given the different implementation challenges. Now, some of these research questions may be relevant also at researcher level. So researchers, when you think of, you are also asked, when you develop a project, you are asked to think of research questions. So you can think in terms of whether your question relates to any of these priorities that have been identified at international level by these quadripartite organizations, because you are supposed to always try to link the work you are doing to SDGs, the Sustainable Development Goals. So in terms of that, maybe this may help you to think how the research that you are doing can contribute to some of these priorities identified by the international organizations. Now coming to, of course I will discuss only one aspect of the antimicrobial susceptibility testing in terms of capacity building. That is because a lot of data generation. Data generation is important. Surveillance, everybody is carrying out surveillance for AMR, and what tools are being used for surveillance. So yesterday Dr. Vandana was saying there is no uniform methodology that has been advocated. Each hospital may adopt their own method, but they may be using different tools like Vitek or other sources of information. So when it comes to susceptibility testing, as we all know, we have a disk diffusion method, there is Kirby-Bauer method, or automated methods like Vitek, or minimum inhibitory concentration method for some of the drugs like colistin. You may have to do you can't do disk diffusion, so you have to go for MIC. And then results are expressed in terms of zone diameter or lowest concentration at which no growth is observed. An important aspect to be considered would be the methodology that you use, because you need to adopt standard methodologies. We heard about standard setting bodies. I think Dr. Vinay also mentioned about CLSI and UCAS. So these are the two major standard setting bodies in terms of standards for determining antimicrobial resistance. So we need to adopt these practices. So they specify what methodology is to be used, because the results that you get will be different based on the type of medium you use, the temperature at which you grow the organism, and so on. Medium and its composition, temperature of incubation, and assessing the performance of tests, use of quality control. So this is again another important aspect to be considered. How do you determine whether an organism is sensitive or resistant? Now, for example, if you look at this plate, you will see, of course, big zone of inhibition here. So you would conclude that, okay, this is sensitive. But here you see there is a zone of inhibition, but this is labeled as resistant. So how do you differentiate a sensitive strain from a resistant strain? It's not just the existence of a zone of clearance, but then to what extent is the clearance taking place? So we need to identify, so we need to strictly follow the methodology of CLSI and UCAS regarding medium ion concentration, temperature growth, and we need breakpoints. So here this is where breakpoints are needed to differentiate sensitive strains from resistant strains. Now these breakpoints have been developed for a number of organisms. So there are two types of breakpoints that are often discussed. One is epidemiological cutoff points, which are breakpoints determined from the analysis of MIC for a large number of isolates. So you need to have large collection of isolates and determine the MIC, and then determine the distribution, and then see at what point most of these organisms are inhibited. So the number of isolates that have particular MIC is plotted to obtain a distribution curve, and the common epidemiological cutoff value for resistance or recoff breakpoint is established as the upper limit of the curve for majority of the population. So among the standard setting bodies, I think UCAS mostly goes in for these epidemiological cutoff points. And then those isolates that have MIC above this breakpoint are considered resistant to the antibiotic, even if the MICs do not reach the clinical breakpoint. So what is this clinical breakpoint? So when you say clinical breakpoint, if we are referring to the level of drug which can be clinically effective. Because in a broth you may find inhibition, but when the drug is taken by a patient, it has to reach the tissue. So at what level it can reach the site of infection, and at what level it can overcome the infection. So that could be different from the epidemiological cutoff point. So you may not have clinical breakpoint data for many of the organisms, except of course in the hospitals you have you may have this. So this illustrates what you get. So you are having this distribution curve here, and then you have the epidemiological breakpoint here, and then the clinical breakpoint may be here. Though the epidemiological breakpoint is here, clinical breakpoint may be different. So this is something that you need to keep in mind when we are talking about breakpoints. Yeah, this is illustrated here. So you can see what you differentiate between the wild type and the resistant ones, and then where do you label them as sensitive. So these organisms up to here are labeled sensitive, and then it's intermediate resistance, and then resistance. So you need data about large number of strains to have this type of breakpoints, and for many of the medically relevant organisms we already have this, and it is being developed by the standard setting bodies like the CLSI and UCAS. But when it comes to aquatic organisms, you may not have all the data, because there are different type of organisms, particularly aquaculture. We are dealing with poikilothermic organisms, so they are cold-blooded animals, so they are growing at low temperature. So FAO came up with this technical paper on the performance of antimicrobial susceptibility testing programs relevant to aquaculture and aquatic organisms. So if you look at the standard setting bodies like CLSI, you have these various organisms listed here. So some of these are relevant for aquaculture, like Aeromonas salmonicida, then Hydrophila, Flexibacterium columnare, Flexibacterium psychrophyllum, then Streptococcus group, Pseudomonas, Listeria. So what is the control strain to be used for these is given in this CLSI guideline. So for Aeromonas salmonicida and Hydrophila you have here. So you need to use these specific organisms as a control strain to determine whether you are achieving the breakpoint and whether you are coming up with an appropriate result. So this is the CLSI guideline, performance standard for antimicrobial susceptibility testing for bacteria isolated from aquatic animals. So now there is a focus on aquatic animals because aquaculture is growing and there is suspected use of antimicrobial, so we need to come up with more information that the aquaculture industry can use to determine resistance. So here you can see the example of E. coli. For E. coli, and what is the disk content and what is the quality control range at the growth temperature of 22 degrees. So you can see it is different when the organism is growing at 28 degrees. So you have to be clear at what temperature the test has been performed and use the appropriate breakpoints that are available. So the guideline gives information like this. For blood dilution, you have the cation adjusted Miller-Hinton broth. For disk diffusion, you have Miller-Hinton agar, and then the growth temperature is mentioned here, and then the organism, the quality control organisms are mentioned here. So we need to go to these standards, but you can see here, this is the table from this standard, but for some, for example, for erythromycin, here this says the breakpoints are not available. So we need to look at some of these aquatic organisms and see whether we can develop some of these breakpoints, because breakpoint development requires a lot of work, coordinated research between several laboratories, hundreds of cultures have to be tested by different laboratories. But there are some publications, like for example this publication talks about epidemiological cutoff values for standard microdilution and disk diffusion susceptibility testing for Aeromonas hydrophila isolated from fish, for Streptococcus agalactiae, and also this is a journal, Disease in Aquatic Organisms, which has called for papers on these ECOFs for aquatic organisms. So that means there is a lot of international interest in generating data. This may be there even in case of veterinary pathogens. I'm sure there will be veterinary pathogens which are not covered under the CLSI normal guidelines. So there is a veterinary standard, so you can look at the organism of interest to you and whether you have the breakpoints that are relevant to the organism that you are working with. And then we'll also look at the whole genome sequencing. Now, of course, everybody talks about whole genome sequencing, and since the cost of sequencing has come down, a lot of people are doing whole genome sequencing and lots of data available. So once you have the whole genome, you can also analyze for the presence of the resistance genes in the sequence. So whole genome sequence can provide new insights to disease transmission. You can know about virulence factors and also about the dynamics of AMR, because it's not just the presence of the gene, whether it is present in a transposable element or it is present in a gene cassette that can be easily transmitted between organisms, and is it related, it is present in a sort of island we call about talk about pathogenicity island or genomic islands, which may be transmitted from one organism to another. So a lot of this kind of information you can get by analysis of the whole genome of the organism. So whole genome sequence can be used to identify and characterize pathogens, and also to trace, say if you isolate from an animal and a clinical isolate, are they same or are they different, how close are they, you can get information on that by using this whole genome sequence. So when combined with epidemiological information, it can facilitate linkages during early detection phase of the outbreak, accurate tracing of transmission chains, and precise delineation of geographical spread. So from where it has come, whether the pathogen in area, one geographical area is same as the one that is present in another, what are the differences. So you can analyze some of these based on this whole genome sequence. So it helps you to identify the sources of infection and timely outbreak detection and removal of sources of infection. So once you have this information, you can try to see how you can eliminate these sources, so which can lead to substantial cost saving in public health. So this whole genome sequence data can be used to verify and identify AMR mechanisms in isolates with relevant phenotypic AMR or with discordant phenotypic AMR. It cannot, however, be used to quantify the level of phenotypic, because sometimes you may find genes, but if you test phenotypically, it may not be resistant, because it may not correlate, there may not be expression of that gene. So you cannot directly translate the genomic information to phenotypic information, so you should always perform both. So just because you have a sequencer and you have the sequence information, it's not that you can do away with the phenotypic method. So AST data could indicate that a high proportion of strains of a bacterial pathogen in a region are resistant to several antibiotics. So this may indicate emergence of a new type of AMR. And the whole genome sequence data can help to understand the location of AMR genes, so where it is present in mobile genetic elements or plasmids or transposons. So this may help in making inferences on what favored the evolution of this organism. Now, but for any policy action or public health action, you need a combination of information. So you need, because whole genome data is here, and then you need clinical data, you need epidemiological data, and then you need susceptibility data. So based on combining the analysis that is coming out from all these different areas, then you can look into what interventions can be suggested in terms of public health and infection prevention and control measures, and what are the community health interventions that can be advised. Because these are just simple examples of nucleotide sequencing. So Sanger method, now of course it is all automated, so you can get these type of sequences quickly, and then you may also have shotgun sequencing where you fragment the DNA and then generate the sequence information, and then you may have NGS, where again you are breaking down the DNA, adding adapters and then sequencing them. So different sequencing platforms are available, so different labs may use different platforms. But once you have this information, then how do you come to a conclusion? How do you analyze? So you have to put together this information. You may have a lot of reads, but then you need to join them to make into fragments and then ultimately the whole genome. So there are different tools that are available for assembling the sequences and making these type of contigs and fragments and ultimately the sequence information. So this summarizes some of the information that the tools that may be used for assembling this genomic information. Different labs may be using different tools. So this is just an example. So if you look at aquatic pathogens, now for example Vibrio parahymorticus is an aquatic organism. It can also cause human infections. It can cause shrimp disease also. So you have 10,000 or nearly 11,000 whole genome sequences available. And one ficus is another serious organism that is transmitted through consumption of live bivalve mollusks. And it is a problem particularly in countries that consume live bivalves like in Europe and in US, and it can cause mortality in 50% of the people, infected people, but it affects mainly immunocompromised people with either liver disease or having high iron content in their blood. Then of course Vibrio agenicus, angularum. So you can see there are a large number of genomic information available. Streptococcus agalactiae. Now this organism has now become important because again agalactiae is present widely in different sources. Earlier it was considered mainly as a source of infection in newborns, but then few years ago there were cases of Streptococcus agalactiae systemic infection in healthy individuals in Singapore, and they traced it to consumption of fish. So now it has become an organism of public health importance. So a lot of sequence information is coming on this Streptococcus agalactiae, and of course it's, I think, also important in cattle causing mastitis and other conditions. So if you look at the various tools available for this kind of analysis, again you have different types of tools available like AMR Finder Plus. Then so there are different types of tools available. So different investigators may use different tools. Now what do you choose, and will you get the same result if you use different tools? So that is something that we need to address. So there are publications like this validating the AMR Finder tools and resistance gene database using antimicrobial genotype phenotype correlations in a collection of isolates. And this type of studies show that there are differences in terms of the output that is obtained from these different analysis. So here they had included over 5,000 Salmonella enterica, 770 Campylobacter, and then there was data from 87,679 susceptibility tests performed. And there was 98.5% consistency, but there were also differences. So the output from one, for example, here you can see the AMR Finder missed 16 loci that ResFinder found, and ResFinder missed 216 loci that AMR Finder identified. So depending on the tool that you use, you may get different results. So you need to think about the method that you are using and what are the limitations that these methods have and how you can overcome these methods. So because you have the comprehensive antibiotic resistance database that is developed, so people are updating this type of database which may provide you more information. So here this is another publication which talks about routine employment of computational tools for AMR resistance determination via high throughput sequencing. So here again you can see some differences in the outputs based on the tools that have been used by these investigators. So you find a lot of this data in the literature, but then you need to see what type of data methodology people have used. This also refers to the phenotypic tests. Now, if you look at the number of publications in aquatic organisms, there has been a large increase. Now, thousands of papers are coming. People are doing susceptibility testing and they are coming to conclusion they are resistant. Now, most of the time they might have not used the CLSI method or the reference train. So you may get results which may not be acceptable. So there has been an analysis how many of these have actually used systematic method using standard protocols. So it is important that researchers are aware about this, so that you can come to the right conclusion about these different tools that are available and how to you can use them. So AMR Finder Plus was developed, but again even with the AMR Finder Plus and the platform they use, there can be differences. So here there's another platform like Abricate. So, but here there can be differences between the different outputs that you get based on the tools that you have used. So some people are trying to work out on what can be the gene flow, the genomics workflow in terms of these identification. So this is one attempt to develop an ISO certified genomic workflow. But then of course people can develop alternatives also. It's not that only this methodology has to be used. So I'd like to summarize saying that phenotypic methods for antimicrobial susceptibility testing have limitations for application of pathogens of aquatic animals because of the lack of standards. So this also could be one of the research topics for students, because you can try to see whether you can work to develop these data that is needed to develop these cutoff points. And then data from whole genome sequencing have immense value in understanding the genetic base of resistance and their location. And that again, the data available of varying quality and bioinformatic platforms for detection of AMR genes have different efficiencies. So a combination of phenotypic and genomic information data along with epidemiological information would be helpful in developing strategies to AMR development and transmission. Yeah, I think that is it. Yeah, thank you very much.